Is weight gain just about calories in, calories out? It's not as simple as you might think. In rare genetic disorders, people gain massive fat even with controlled calories. Why? Well, hormones, especially insulin, are calling the shots. Leptin, POMC, and MC4R gene mutations all lead to one thing: chronically high insulin. And insulin, of course, is the master switch for fat storage. These genes disrupt brain signals that control hunger, energy use, and how much insulin your pancreas makes, even altering fat storage at the cellular level. Obesity isn't just about overeating, it's about energy partitioning, and hormones decide where that energy goes. This is lecture 114 of the Metabolic Classroom. Looking to improve your own metabolic health? Visit insuliniq.com for courses, coaching, consultations, and a 10-day free community membership trial. To dive deep into the science behind metabolic health, become an insider at benbikman.com, where you'll enjoy my exclusive content, ad-free podcasts, live stream Q&A access, and more. Welcome to the Metabolic Classroom. I'm Professor Ben Bikman, a biomedical scientist and professor of cell biology. Today, we're covering a topic that reveals just how incomplete the calories in, calories out model really is. And we do so by exploring monogenic obesity. These rare genetic conditions offer a window into the true drivers of fat accumulation, and they underscore the central role of hormones, something you know I am always interested in doing. Most people have heard the standard model of obesity: eat more calories than you burn and you'll gain weight. It's certainly neat and tidy and simple, but it is also incomplete. Yes, calories matter. They are the fuel that gets stored, but what determines whether those calories are burned for energy or stored as fat is more than just a math equation. It's in fact endocrinology. It's hormones. Hormones act as chemical instructions to our cells, dictating whether to store fuel or use it. And there's one hormone that, of course, ultimately decides whether fat cells grow. It's the one hormone to rule them all, and that is insulin. Without elevated insulin, fat cells simply will not expand. Insulin signals fat cells, also called adipocytes, to take in glucose and convert those into fats, or just pulls in fatty acids, but ultimately converting them into triglycerides, the stored form of fat. And then it locks them away while fat release, it blocks the fat release, rather. So not only is it facilitating the uptake and storage, but then it's preventing them from breaking down by inhibiting lipolysis. Now, as I noted, to illustrate this in the relevance of hormones, I want to explore monogenic obesity. These are the rare cases where a single gene has been mutated. That's why it's monogenic, and it then causes profound obesity. Now, no surprise, insofar as this is a genetic problem, it often manifests in infancy or very early childhood. And this is not your run-of-the-mill common form of obesity influenced by lifestyle. They're really driven by this very specific genetic defect. But they do reveal the inner workings of our inherent weight regulation system, and it shows why a calorie-only explanation just cannot fully explain human obesity. In these conditions, even when calorie intake is controlled, as you'll see, fat storage nevertheless ramps up, and it's because of the disrupted hormone signaling. And we'll all kind of bring that back to a singular hormone, as I've already alluded to. Now, today, we'll walk through the hypothalamic POMC pathway, which is a key brain circuit for appetite and metabolism. And we're going to examine three major genetic defects within it: leptin and the leptin receptor, POMC deficiency, and MC4R mutations. So we'll explore and explain all of those. But we'll see how each one disrupts energy balance and ultimately affects insulin, of course, which collectively reinforces this heavy fat storage phenotype. And I'll draw on, of course, as ever, peer-reviewed evidence from both animal and human studies, just so that you're comfortable knowing that there's no speculation here, just a lot of science. All right, let's start with the basics of the hypothalamic POMC pathway, because understanding this circuit is crucial to grasping how these genetic mutations work. Deep within the hypothalamus, which is a small region of the brain that has a lot of influence in a lot of processes, but it acts as a master regulator for things like hunger and body temperature and controlling a lot of hormones. But within that section, within the hypothalamus, is the arcuate nucleus. This area houses two main sets of neurons that oppose each other in function. One group, the AGRP or agouti related peptide, and the NPY, neuropeptide Y neurons, drives appetite and promotes energy conservation, essentially telling your body to eat more and burn less energy. Now, the other signal, remember I mentioned there were two. So there was the AGRP and the NPY, but then the other one is the POMC neurons. POMC stands for pro-opiomelanocortin. Pro-opiomelanocortin, POMC neurons. And those suppress appetite and boost energy expenditure. So that would be an instance of you feeling more satiated or full, and your body's using energy more readily. POMC neurons are particularly fascinating. They produce a large precursor protein called, no surprise, pro-opiomelanocortin. So that's the protein product. And this then gets split or cleaved into smaller active peptides. The most important for metabolism is alpha melanocyte stimulating hormone, or alpha MSH. Alpha MSH then binds to and activates melanocortin 4 receptors, MC4R, on these downstream neurons. And that then sends a signal of enough energy, stop eating, and burn more. But the pathway doesn't just control food intake. It responds to hormonal inputs like leptin, which is coming from fat cells, insulin coming from the pancreas, and even famous gut peptides like GLP-1. It then sends an output through the autonomic nervous system. That's the part of the automatic controlling of the body. And the autonomic part is more involved in the rest and digest part of our body, as opposed to the sympathetic aspect and the parasympathetic, or playing into the autonomic. So the autonomic is actually is overseeing both of those, the sympathetic and the parasympathetic. And it sends these signals throughout the body to the liver to adjust glucose production, to the muscles, and even the pancreas, which is something, of course, that's important because of its relevance with insulin. When leptin, for example, activates POMC neurons, several things will happen. Appetite will drop, energy expenditure will go up, in part through thermogenesis, like brown fat tissue. Liver glucose production will go down, which helps control blood sugar, and muscles and liver will be more insulin sensitive. All of this keeps energy partitioning balanced. It is, in fact, favoring the burning of energy rather than the storing of energy. But if leptin signaling is disrupted, these effects, of course, are broken, if you will. Then the person is going to be eating more. There's going to be less energy expenditure, less insulin sensitivity, and in fact, a poorer regulation of insulin secretion from the pancreas. Okay, now I've sort of already been getting into leptin a little bit, but let's actually now turn to leptin very directly. And we'll say that this is the first genetic defect that we'll explore. And it's both leptin production or leptin receptor deficiency. So that's very important to appreciate, that in both instances, you can have a mutation in leptin, but it could be on the front end. In other words, you don't make leptin, or it could be a problem on the back end. In other words, you don't make the receptors for leptin. So there's a receptor mutation or a leptin itself mutation, but the end result will be identical. This condition, of course, disrupts the body's ability to sense energy stores. So this is a signal coming from the fat cell. And in the absence of that signal, the brain is left thinking, well, there are no fat cells. This then leads to, unsurprisingly, unchecked appetite and an energy situation where the body really, really wants to store energy. Now, leptin has an interesting history. It was identified as a hormone in 1994, so well within recent memory, but it actually was relying on animals that had been studied, what's called an OB mouse, OB just standing for obesity, and the fact that I just mentioned two of them, OB and OB, means that the animal got the gene from both mother and father. So it's monozygous. It has the same gene there. So the OB mice had been used for decades previous to this. So in fact, some of the evidence I'm referring to throughout this discussion comes from studies published before leptin was called leptin, as it was identified formally in 94. We knew about the animals and scientists had been studying the animals. But leptin is a hormone, as I've talked about abundantly in the past, that comes from fat cells. And typically it's coming in proportion to fat mass. So if a person has more fat mass, they make more leptin. And then leptin acts on leptin receptors that are found throughout the body, but especially in the hypothalamus at those POMC neurons. And that becomes the central brain signal that the energy stores are adequate and there's no need to overeat. And there's no need to hold on to energy or go into conservation mode, that we can spend it, that we have plenty of energy. Now, in congenital leptin deficiency, or in the leptin mutation, the hormone is absent because of the lack of the LEP, or the leptin gene. But then on the other end, you can, of course, have the mutation being in the leptin receptor. But again, in both cases, the hypothalamus interprets this as starvation. Appetite surges via unchecked activation or activity of that pathway I mentioned earlier, the agouti-related peptide or neuropeptide Y, so AGRP and NPY. This then leads to hyperphagia, or just uncontrolled hunger. And this is something, as a scientist who has worked with OB mice and the rat counterparts, they are insatiable. There is just an uncontrolled hunger. But leptin's role, it is very important to understand that it goes beyond just hunger. Classic experiments with the OB mice and the rat equivalents show that even when calories are matched, let me say that again, in leptin mutant animals, even when they're eating the exact same amount of calories as wild type or non-mutant leptin mutant mice, fat accumulation is still higher. So this is a process called pair feeding, where you only allow one group of animals to eat the same amount that another group of animals are eating. And pair-fed leptin deficient mice, so given the exact same calories as control mice, still develop more body fat. And this is, in fact, potentially quite dramatic. In one study, leptin deficient mice stored nearly 4 times more fat compared to the control counterpart mice, even though they ate the exact same number of calories. And this has, of course, been echoed to varying degrees in other work. That study I mentioned was from the 1970s. There's been other studies to show it, and in fact, in some instances, even more dramatically. But critically, in the absence of leptin signaling, we have a host of peripheral effects. As I mentioned, leptin affects a lot of tissues that go just beyond the brain. Skeletal muscle is using less glucose, brown fat thermogenesis goes down, insulin sensitivity drops. Interestingly, the pancreas then comes in here as well by secreting a lot more insulin, which is critical for fat storage. In essence, the lack of leptin signaling, yes, it influences hunger. As I noted, these animals have an insatiable appetite, and the human equivalents do too. But it just simply reprograms the overall metabolic status of the body, that more fat is stored per calorie consumed. That's critical. And it just kind of challenges just the simplicity of calories in, calories out. Hormones determine how that energy gets stored or used, whether it's burned or wasted in the form of ketones or heat. All right, well, that's enough for leptin. I think I've painted that picture adequately, clearly. Let's go on to POMC deficiency. So we're, if you will, moving downstream in the pathway and looking at the lack of POMC. This disrupts the production of alpha MSH. That's the melanocyte stimulating hormone that I mentioned earlier. And this leads to a lack of satiety and reduced insulin sensitivity, with some other changes in insulin secretion from the beta cell. POMC deficiency arises from mutations in the POMC gene, which then halts the production of alpha MSH entirely. Without alpha MSH, MC4R, the melanocortin 4 receptor, isn't activated. The activation fails, leading to this absent satiety signal and a reduction in energy expenditure. These deficiencies actually also disrupt adrenal function, because ACTH, adrenocorticotropic hormone, is another product of POMC. So remember earlier I mentioned that POMC is this, it becomes a mother molecule that gets cleaved or split into more active parts. I've only been mentioning the alpha MSH, but the other part is ACTH. And ACTH is essential for cortisol production. That's, of course, coming from the adrenal cortex, which is very important. Don't ever think of cortisol as a negative, as a villain, because we call it a stress hormone. It is essential in helping maintain blood glucose. It regulates metabolism. It regulates immune responses. Yes, too much is too much of a good thing, if perhaps we could say, but it still is important. But anyway, to bring it back to POMC, if you don't make any POMC, yes, you're not making alpha MSH, but you're also not making ACTH. So this can result in a lack of adrenal function, what we could accurately call adrenal insufficiency, where the body can't produce enough cortisol. But metabolically, the loss of alpha MSH is pivotal. The melanocortin system fails to restrain feeding and enhance peripheral insulin sensitivity. And this reduced sensitivity, of course, not only is causing insulin resistance, but also contributing to hyperinsulinemia. As an interesting aside, and I say this as a former redhead, I know you can't tell, all my former glory is gone at the moment, but it is an interesting aside that this loss does cause a quirky change in pigmentation. Without alpha MSH, a different melanocortin receptor, MC1, is understimulated. And MC1 is responsible for creating a melanin that is a brown pigment. But in the absence of stimulating MC1, the person ends up creating a lot, in fact, exclusively, pheomelanin, which is the melanin that is a red-yellow pigment. And the result of this is someone with bright red hair and, in fact, very, very pale skin. So it's an interesting feature that if someone has the POMC mutation, yes, they will invariably become obese and they will invariably have red hair. In both human and animal models, POMC disruption will contribute to hyperinsulinemia. And again, we're going to come back to that idea in just a moment at the end to just bring it back to one singular hormone. But the relevance of this in animals is very, very clear. In pair feeding experiments, just like I mentioned with leptin a moment ago, the POMC deficient animals will still gain significantly more weight and fat mass, often double the fat mass of the control animals. Again, even though they are eating the exact same number of calories. And this just further highlights the metabolic shift toward storage that has nothing to do with how many calories the animal is eating. In humans, a patient with complete POMC deficiency will have all of the features you'd expect. Massive obesity, yes, bright red hair, and all the metabolic consequences that come from the obesity and the over secretion of insulin. In the POMC deficiency cases highlight just the consistent early onset obesity, the hyperphagia, so the uncontrollable appetite, and the hypocortisolism, which is going to make it very hard for the patient to fast. Just telling a person with POMC to eat less, that in fact not only would be fruitless, but it could in fact be harmful. And all of this is just because in the absence of these signals with this mutation, calories are just not partitioned well. Because of the disrupted melanocortin signaling, it reduces peripheral insulin sensitivity, it stimulates insulin secretion, and ultimately we have a body that just is determined to store energy at all costs. All right, now moving on from POMC, let's get to the last part of this, which is the MC4R mutations. They're actually the most common form of monogenic obesity, which is to say it's uncommon, but it's about 1% of very obese individuals. So 1% of these individuals have this most common monogenic form of obesity. This defect weakens satiety and energy expenditure, while once again impairing insulin sensitivity and leading to hyperinsulinemia. MC4R is the receptor for alpha MSH, which means it is downstream of POMC, which we just spoke about. So we have the POMC neurons producing POMC, splitting into alpha MSH and ACTH. Alpha MSH is the protein of interest here because it binds to and activates MC4R. But what if MC4R is mutated, which again is what we're discussing now? Defective MC4R weakens or abolishes the satiety signal entirely. So that means appetite is increased or insatiable, and it is reducing energy expenditure throughout the body. Now, once again, beyond the hyperphagia or the uncontrollable eating, MC4R deficiency once again connects to hyperinsulinemia. The clinical spectrum of MC4R mutations in obese patients shows early onset, no surprise, increased fat mass, elevated insulin levels, even adjusting for BMI. Rodent models confirm this. If you have an MC4R knockout mouse, they have very, very high insulin, and they will then start to become very obese. So the hyperinsulinemia is going to is predates or is a prelude to the significant weight gain. In youth, rare damaging MC4R variants are linked to greater insulin resistance, visceral fat, and intrahepatic fat, even when looking at just BMI. A homozygous MC4R mutation in a Japanese woman, for example, is a case study. She presented with severe obesity and hugely, massively high levels of insulin. In pair feeding experiments in animals, the MC4R deficient mice will gain up to about 2.5 times more fat than the control animals, even when eating the exact same number of calories, demonstrating once again that obesity isn't just from overeating, but a deviation or a disruption in energy partitioning. These data certainly suggest MC4R is helpful for insulin sensitivity. It helps regulate the sympathetic outflow of the autonomic nervous system. And in that sympathetic side, as I kind of bumbled through it in the intro, is the fight or flight part. And when that's activated, it's bringing down insulin production. So, of course, with this mutation now, we've removed this brake. All right, now to start to wrap it all up, these three conditions, leptin, or the leptin receptor mutation, POMC deficiency, or MC4R mutations, all start differently. But as you've been gathering throughout this mini lecture, they all converge. And that convergence, that nexus, is chronically elevated insulin. And that is key. Insulin directly signals fat cells to store triglycerides and inhibit lipolysis. The pair feeding experiments across these models prove you can eat the same number of calories yet gain dramatically more fat if insulin is high and partitioning is altered. So as much as we look at these problems, and too often people will say, well, a leptin deficiency or POMC or whatever they're fat just because they're overeating. They can't control their appetite. Yes, in a real world setting, they will not be able to control their appetite, or it's going to be inhumanly difficult to do. But even when you control for calories, we see this. So a calorie-only obesity model ignores this. The calories provide the material, the building blocks, but the hormones instruct the use of those building blocks. And insulin is the final authority when it comes to fat storage. In fact, just to put a fine point on that, to understand how the genetic defects elevate insulin, I may as well just elaborate the mechanisms here. With leptin deficiency, it's important to appreciate that leptin elicits an inhibitory signal on the beta cells to reduce insulin. So in the absence of leptin, it removes, we've lost this inhibitory effect on the pancreatic beta cells, where insulin will normally bind to where leptin will normally bind to the receptors to suppress insulin secretion and gene expression. So the consequence is unchecked insulin production. In fact, Dr. Jeff Friedman, who identified leptin, in exploring one of his off-the-record quotes that Gary Taubes has identified in his reporting work, he says that insulin goes through the roof. That was his quote, that insulin goes through the roof in the animals that lack leptin. For POMC deficiency, the disruption in hypothalamic POMC neurons alters the autonomic flow or signal, including increased parasympathetic vagus nerve activity to the pancreas. So that's it's a direct line. That vagus is a direct line from the hypothalamus to the pancreas and the beta cells. And when the POMC signal is disrupted, it is just an incessant go, go, go signal to the beta cells. So the result, no surprise, is a huge amount of insulin release. And similarly, MC4R mutations prevent alpha MSH from activating MC4R in the vagus, which once again results in heightened vagal tone that stimulates insulin coming from the beta cells, so a direct signal. So regardless, every form of monogenic obesity, the nexus is insulin. There's no getting around insulin's relevance here. So in closing, monogenic obesity teaches that obesity isn't always just a matter of willpower, and it's just a matter of arithmetic. There's biochemistry, there's endocrinology here, that if the hormones dictate fat storage, every calorie consumed is going to be more inclined to be stored as fat rather than burned. I hope these thoughts are helpful. It certainly, I hope, provides some nuance on the conversation of just why we get fat. It's not just calories. Until next time, more knowledge, better health. Looking to improve your own metabolic health? Visit InsulinIQ.com for courses, coaching, consultations, and a 10-day free community membership trial. To dive deep into the science behind metabolic health, become an insider at benbikman.com, where you'll enjoy my exclusive content, ad-free podcasts, live stream Q&A access, and more.